Cystic fibrosis is an autosomal recessive disorder that affects 70,000 people worldwide. This is due to mutations related to the cystic fibrosis transmembrane conductance regulator, or CFTR, which is found in multiple organs, in particular the respiratory tract. The CFTR channel allows movement of chloride ions in and out of cells, which is important for salt and water balance. Cystic fibrosis therefore causes abnormal ion transport. As a result, cystic fibrosis is mostly characterized by thick mucus buildup in the lungs and can also greatly impair functions of the pancreas, liver, and reproductive organs, hence putting patients at a great health risk and reducing their quality of life.
Classes of CFTR mutations that can result in decreased chloride conductance. The Class 1 mutation is characterized by insufficient mRNA translation caused by a premature stop codon resulting in decreased protein expression. In Class 2, there is a defective protein folding of CFTR resulting in decreased trafficking to the membrane. This is due to deletion of DNA sequences such as delta F508, causing CFTR to remain in the endoplasmic reticulum where it is degraded. In the third class of mutations, CFTR is trafficked to the apical cell membrane but fails to transport chloride due to a defective gating mutation. Much like Class 3, the Class 4 mutation results in a misshaped CFTR pore, which decreases chloride permeability through the channel. In the fifth class of mutations, there is a reduction in the CFTR protein due to incorrect gene splicing prior to transcription. Lastly, in Class 6 mutations, the CFTR channel has an abnormally short residence period at the apical cell membrane due to increased turnover of the protein at the cell surface.
Current therapies for cystic fibrosis include treatments such as anti-inflammatories, antimicrobials, bronchodilators, and medications assisting with mucociliary clearance. Beta-2 agonists are also used to decrease plasma exudation in the lungs by targeting respective receptors. Other CFTR-specific therapies include drugs such as lumacaftor and ivacaftor, which have also proved to be effective in recent years.
In cystic fibrosis, Class 2 mutations affect almost 90% of sufferers. This is why our team at CPC Pharmaceuticals have recently developed a novel therapy to target this class of mutation. Our treatment involves encapsulating an mRNA with a transcription export complex, or Trex, within a nanoparticle. The mRNA strand corresponds to a healthy CFTR gene sequence. The nanoparticle will be transported into the respiratory epithelial cells to substitute the patient's delta F508 mutation.
The nanoparticle will be composed of silica coated with TPGS and albumin, which will assist diffusion into the cells down the concentration gradient. The TPGS enhances the delivery of our nanoparticles through the mucus layer, whilst the albumin coat improves uptake into the epithelial cells. Once inside the cytoplasm, our nanoparticle will be manufactured to respond to the cytoplasmic pH and release the mRNA. The Trex complex will facilitate the transport of mRNA to the ribosomes. The ribosomes will translate our RNA into a fully functional CFTR protein, which will be trafficked to the cell membrane and allow for proper chloride transport.
Challenges currently present in treating cystic fibrosis are due to the multifactorial origins of the disease and the variety of different mutations of the CFTR gene. This makes it harder to target each type of cystic fibrosis with the same treatment. Cystic fibrosis also affects multiple organs, which also increases the difficulty of developing a treatment which can target all these areas.
To test whether our treatment will work in vitro, we will use an immortal cell line that expresses CFTR. By knocking off the gene for CFTR and then administering our nanoparticle with mRNA, we can see if our treatment is actually effective. We can also tag the nanoparticle and the mRNA with a fluorescence so we can visualize how long and where the nanoparticle travels and when the mRNA is released.
Now we have chosen ferrets for our preclinical trials. Ferrets have a 91% similarity to human CFTRs. They have a short gestation period and a short sexual maturity duration, and this will allow an efficient, cost-effective treatment for CFTRs. Soon enough, we will be able to develop a suitable and effective treatment for clinical trials within the near future.
The nanoparticles could also be used in a percutaneous injection into the liver. Cystic fibrosis-related liver disease affects 30% of patients with cystic fibrosis. It damages the hepatobiliary system because secretions are far too thick and acidic. The fluid builds up in the secretory ducts, which impairs nutrient absorption and causes damage. Therefore, liver pathology will cease and self-repair will occur.
Our team here at CPC Pharmaceuticals is very dedicated to finding a cure to cystic fibrosis. Please support us in bettering the lives of thousands of people.